Hello, I'm Husayn El-Sharif with the Georgia Water Resources Institute at Georgia Tech, and this is our presentation on satellite-based assessment of Lake Lanier Chlorophyll A. This research was partially sponsored by Gwinnett County Department of Water Resources and by the Georgia Water Resources Institute at Georgia Tech. My co-authors are Xiaofeng Liu, a doctoral student at GWRI, and Professor Aris Georgakakos, the GWRI director. To begin, we take advantage of fine spatial and temporal resolution satellite data to monitor the water quality of Lake Lanier. Lake Lanier is a vital freshwater resource for both the city of Atlanta and the state of Georgia as a whole, providing drinking water, flood control, navigation, hydroelectric power, recreation, and environmental management. Chlorophyll A is a pigment inside of phytoplankton, which is widely used as a proxy for algae biomass and water quality in water bodies. Excessive amounts of algae in water typically indicate high influx of nutrients, such as nitrogen and phosphorus, which can cause lakes to become eutrophic and which pose risks for both the environment and human communities. Chlorophyll A reflects, emits, and scatters radiation in a characteristic manner, and this behavior can be sensed by satellite systems, such as the Sentinel-2 satellite system, which we use in this work, in order to derive photic-zone chlorophyll A concentrations. So the objective of this study is twofold. Firstly, we want to develop robust relationships between in situ or field measured chlorophyll A, which are depth averaged through the photic zone, and Sentinel-2 imagery. And secondly, we want to use these relationships and satellite images to develop a user-friendly operational tool for assessing water quality over the entire lake, not just those regions of the lake where measurements were taken. The first part of this project involved an extensive field sampling mission in Lake Lanier, carried out from the fall of 2018 and completed in the fall of 2020. So at multiple locations throughout the lake, our team measured chlorophyll A along with turbidity, temperature, pH, and so on at multiple depths. The field sampling dates were also scheduled to coincide within a few days of the Sentinel-2 satellite overpass. Focusing on chlorophyll A, we attained both chlorophyll A measurements in the field as well as water samples that were later analyzed in the labs of Gwinnett County, in order to get the most accurate characterization of chlorophyll A in the lake. Next, we linked together depth average chlorophyll A measurements with multi-band satellite retrievals from the Sentinel-2 mission. About Sentinel-2, it is a 2-satellite system launched by the European Space Agency in 2015, and it is still operational. The satellite constellation measures top of atmosphere reflectance from 13 spectral bands in the visible, near infrared, and shortwave infrared parts of the spectrum at spatial resolutions ranging from 10 meters to 60 meters. The satellites pass over Lake Lanier around noontime about twice a week. So using our field data and various regression models, we identified the Sentinel-2 bands and band ratios that had a strong connection to photic zone chlorophyll A measurements. And those bands and band ratios are indicated here. It should also be noted that there were some challenges to overcome in working with the Sentinel-2 data. In order to develop the robust relationships between field measurements and the Sentinel-2 bands, we needed to work with the most pristine satellite images, as indicated here. And those are images that are free from cloud interference and also free from sun glint. In our analysis, we found that we could automate the process of filtering out both cloud and sun glint contaminated images by monitoring the reflectance measured in the shortwave infrared band, or the band B12 here, measured over the lake. After going through this filtering process, we end up with about 2 to 3 clean or pristine images per month. And also, to avoid noise interfering with the retrievals, we also aggregate the Sentinel-2 pixels to the 30-meter spatial resolution. Here are the results of our models which link the Sentinel-2 bands and band ratios to photic-zone chlorophyll A. Based on the thermal properties of the lake, we divided the year into 3 seasons, namely the November to April, May to July, and August to October. And we also looked at what were the best linear and non-linear relationships between the Sentinel-2 data and the depth averaged, or the photic-zone chlorophyll A measured in the field. The average error between the observed and modeled predicted chlorophyll concentration has a R-squared statistic ranging from 0.7 to 0.9 and a root mean squared error of about 1 microgram per liter. Here we apply our model to every 30-meter aggregated satellite pixel over Lake Lanier. These two images show the summertime peak in the photic zone chlorophyll A concentration, as well as the wintertime lows. And these results also suggest where we should focus new sampling missions by taking a look at any places or regions or coves that have higher chlorophyll A concentrations. Here we have the time series plot, which shows the photic zone chlorophyll A averaged over the entire Lake Lanier, and it is a convenient way to keep track of how chlorophyll A varies over time. Again, we see clearly how concentrations change from month to month and year to year, with lake-wide chlorophyll A exceeding 8 micrograms per liter during the summer and dropping to below 4 micrograms per liter during the late fall and winter. As time goes on and as more years are analyzed, such sequences could indicate whether water quality conditions are remaining stable as we go from year to year or are deteriorating. Here's another example of chlorophyll A estimated on February 25th, 2021. It's one of the more recent images. From this image, we get a sense of the dynamic range of chlorophyll A throughout the lake. And we see that for the lake as a whole, that the concentration of chlorophyll A is relatively low. However, there are some coves with heightened concentrations that are approaching 10 micrograms per liter. And these locations are places where we may be interested in taking a closer look with targeted field sampling campaigns. So this tool with these maps has a number of useful applications, such as alerting for algal blooms, or identifying regions that are hot spots or nutrient-rich areas, which again we may want to target for field sampling. And we can combine these maps with hydrodynamic models in order to forecast algae blooms and of course assess the lake eutrophic status, which changes over time. We also, for now, this study focused on chlorophyll A, but the same research that goes into monitoring chlorophyll A concentrations can be also used to monitor cyanobacteria concentrations, or the blue-green algae, if more field data becomes available. In this image here, we see how our satellite-based assessment tool can be used to provide algal bloom alerts. On the weekend of October 10th, 2020, after a series of storms and a sewer spill, an algae bloom was spotted in the Flat Creek. We applied our chlorophyll A model to the Sentinel-2 overpass, which were able to get a pristine or clean image on October 13th, and we identified heightened chlorophyll A concentrations in the Lake Cove, where Flat Creek enters Lake Lanier, which is shown here. We have these heightened concentrations relative to the rest of the lake. And we identified these heightened concentrations while field samples were still being analyzed. After developing the satellite-based model for Lake Lanier, we had the question of whether the model would work for other similar lakes in the state of Georgia. So we also carried out a preliminary assessment over Lake Allatoona, and we found the results to be encouraging. We expected that Lake Allatoona would exhibit somewhat higher concentrations due to its smaller size and assimilative capacity relative to Lake Lanier, and indeed our results were consistent with these expectations. So we have here, I have put some images here that compare Lake Allatoona and Lake Lanier. So these two images are from January 25th, 2019. And we see from the, at least qualitatively from the images, that the Lake Lanier is more pristine, while the Lake Allatoona seems to have some more impaired water quality. And that's what's shown in here, that we see the large discrepancy between Lake Lanier and Allatoona, with the Allatoona having the higher estimated chlorophyll A concentrations. And then likewise later in the year, in the fall time, we see this image from September 17th, where the two lakes appear to, at least qualitatively, appear to look the same with respect to the chlorophyll image, or the chlorophyll concentration. And that's what plays out. That also is what plays out here. Yes, here, where they have a similar, both lakes have a similar chlorophyll A concentration. So we want to do a further analysis with this, and this would require to actually have more field samples from Lake Allatoona, but the results here are again encouraging, and this also shows that this model may have regional, or has regional applications throughout the state of Georgia. Lastly, this slide here shows the information technology platform which hosts our satellite-based assessment tool. A major component of this platform is the Google Earth Engine platform, which provides access to satellite data and also facilitates various data analyses through a simple web browser. Using the Google Earth Engine, we have automated the process of the Sentinel-2 data acquisition, image filtering, chlorophyll A estimation, and the creation of chlorophyll maps. And this is just an example here of the Google Earth Engine interface, which is based off the JavaScript language, which again can access the Sentinel-2 data and perform the analyses that were shown in this study. In conclusion, our satellite-based algorithm provides fine resolution estimates of photic-zone depth average chlorophyll A, which is a critical water quality indicator. The model can be applied not just for Lake Lanier, but for lakes throughout the state of Georgia, and this tool provides an easily accessible means to issue early warnings for harmful algae blooms, provide guidance for field sampling missions, and also provides a way to monitor regulatory compliance. We would like to acknowledge our project sponsors, which are the Gwinnett County Department of Water Resources, as well as the Water Resources Institute at Georgia Tech. Thank you for your attention.